शुरू अल्लाह के नाम से जो बड़ा मेहरबान निहायत रहम करने वाला है। अस्सलाम वालेकुम स्टूडेंट, सैफुल्लाह here from Chemistry Department, University of Education, Lahore. Today we are going to discuss the topic is specificity of immunoassay and its different types. Course name is Analytical Chemistry and the course code is CHEM2112. This lecture would help the student to understand immunoassay and its function, importance of immunoassay, fluorescence immunoassay, and last one is the radio immunoassay and its importance. Immunoassay. An immunoassay is a biochemical test that measures the presence or concentration of macromolecule or a small molecule in a solution by the use of an antigen or sometime antibody. The molecule detected by immunoassay is often referred to as an analyte and is in many cases a protein, although it may be other kind of molecule of different size and types as long as the proper antibodies that have the adequate properties for assay are developed. Analytes in biological liquid such as serum or urine are frequently measured using immunoassay for medical and research purposes. Immunoassay come in many different formats and variations. Immunoassay may be run in multiple steps with reagents being added and washed away or separated at different points in the assay. Multi-step assay are often called separation immunoassay or heterogeneous immunoassay. Some immunoassay can be carried out simply by mixing the reagent and sample and making a physical measurement. Such assays are called homogeneous immunoassay or less frequently known separation immunoassay. The use of a calibrator is often employed in immunoassay. Calibrator are solutions that are known to contain the analyte in the question, and the concentration of this analyte is generally known. Comparison of an assay response to a real sample against the assay response produced by the calibrator makes it possible to interpret the signal strength in the term of presence or concentration of analyte in the sample. In some cases, an immunoassay may use an antigen to detect for the presence of antibodies, which recognize that antigen in a solution. In other words, in some immunoassay, the analyte may be an antibody rather than an antigen. Classification and formats of immunoassay. Immunoassay can be run in a number of different formats. Generally, an immunoassay will fall into one of several categories depending on how it is run. Competitive homogeneous immunoassay. In a competitive homogeneous immunoassay, unlabeled analyte in a sample competes with labeled analyte to bind an antibody. The amount of labeled unbound analyte is then measured. In theory, the more analyte in the sample, the more labeled analyte gets displaced and then measured. Hence, the amount of labeled unbound analyte is proportional to the amount of analyte in the sample. Competitive heterogeneous immunoassay. As in competitive homogeneous immunoassay, unlabeled analyte in a sample competes with labeled analyte to bind an antibody. In the heterogeneous assay, the labeled unbound analyte is separated or washed away, and the remaining labeled bound analyte is measured. One site non-competitive immunoassay. The unknown analyte is the sample bind with labeled antibodies. The unbound labeled antibody are washed away and the bound labeled antibody are measured. The intensity of the signal is directly proportional to the amount of unknown analyte. Two-site non-competitive immunoassay. The analyte in the unknown sample is bound to the antibody site, then the labeled antibody is bound to the analyte. The amount of labeled antibody on the site is measured. It will be directly proportional to the concentration of analyte because the labeled antibody will not bind. If the analyte is not present in the unknown sample, the type of immunoassay is also known as sandwich assay, as the analyte is sandwiched between two antibodies. For over 40 years, immunoassay have been used in hospitals, laboratories, different medicine, and research to improve the health and well-being of humans and animals. Information gained by clinical immunoassay testing has shortened hospital stay and decreased the severity of illness by identifying and assessing the progression of disease, thereby leading to improved the therapeutic choice. In life science research, immunoassays are used in the study of biological system by tracking different protein, hormone, and antibodies. In industry, immunoassays are used to detect contaminants in food and water, and in quality control to monitor specific molecules used during product processing. Immunoassays are quick and accurate tests that can be used in site and in the laboratory to detect a specific molecule. Immunoassays rely on the inherent ability of an antibody to bind to the specific structure of a molecule. Antibodies are proteins generated by the animal in response to invasion of a foreign molecule antigen into the body. Antibodies are found in the blood and tissue fluid and will bind to the antigen whenever it is encountered because antibodies are developed to the specific three-dimensional structure of an antigen or analyte. They are highly specific and will bind only to that structure. Once purified from the blood, monoclonal and polyclonal antibodies are ideal assay reagent to detect and monitor specific target molecule with the limited interference from other substances. Four types ELISA formats are monoclonal, polyclonal, sandwich assay, competitive inhibition assay, antigen down immunoassay, and rapid assay. Monoclonal, polyclonal, sandwich immunoassay. In a typical macrotiter plate sandwich immunoassay, a monoclonal antibody is adsorbed on the plastic macrotiter plate. When the test sample is added to the plate, the antibody on the plate will bind the target antigen from the sample and retain it in the plate. When a polyclonal antibody is added in the next step, it is bound to the target antigen already bound to the monoclonal antibody on the plate, thereby forming an antigen sandwich between the two different antibodies. This binding reaction can then be measured by radioisotopes, as in a radioimmunoformate, or by enzyme, as in a enzyme immunoformate ELISA, attached to the polyclonal antibody. The radioisotopes or enzyme generates a color signal proportional to the amount of target antigen present in the original sample added to the plate. Depending the immunoassay format, the degree of color can be detected and measured with the naked eye as with the home pregnancy test. Now we discuss the antigen down immunoassay. In an antigen down immunoassay, the analyte is coated onto the 96-well macrotiter plate rather than an antibody and used to bind the antibodies found in a sample. When the sample is added such a human serum, the antigen on the plate is bound by antibodies from the sample, which are then retained in the well. A species specific antibody labeled with HRP is added next, which bind to the antibody bound to the antigen on the plate. The higher the signal, the more antibodies there are in the sample. Antigen down assay can be configured as a rapid test and are often used to diagnose allergy condition. Routinely, patient blood is tested against different allergens to see if the person has antibodies to that allergen. Competitive inhibition immunoassay. In addition to monoclonal, polyclonal antibody sandwich format, many immunoassays are structured in a competitive inhibition format. Competitive inhibition assays are often used to measure small analytes because competitive inhibition assay only required the binding of one antibody rather than two. As in a standard ELISA format, because of high probability of steric hindrance occurring when two antibodies attempt to bind to a small molecule at the same time, sandwich assay format may not be feasible. Therefore, a competitive inhibition assay would be preferable. Rapid immunoassay. In addition to macrotiter plate, immunoassay are also configured as rapid test. Radioimmunoassay. Radioimmunoassay work by using radioactivity labeled antigen or antibodies. Radioactivity emitted by the bound antibody antigen complex is detected and recorded. Fluoroimmunoassay. Fluoroimmunoassay work by using antibodies that have been labeled with fluorescent probes. Fluorescence from the antibody antigen complex is detected and recorded. Chemiluminescent immunoassay. Chemiluminescent immunoassay work in a similar way to the ELISA, but they measure the light emitted from a chemical reaction. Counting immunoassay. Counting immunoassay used polystyrene beads that are coated with the multiple antibodies complementary to the target antigen. An antigen is a molecule that is recognized by the immune system, particularly by antibodies, protein, polysaccharide, pesticide, antibiotics, toxin, and hormones all can be antigen. But not all antigen can be stimulate the immune system to generate antibody. An antigen that can elicit an immune response and particularly antibody synthesis is called an immunogen. Protein with molecular weight higher than 5 to 10 K are immunogen, while small peptide, pesticide, antibiotics and hormone are not. These low molecular weight substances are called heptene and must be chemically coupled to larger carrier molecule such as bovine serum, albumin. An antibody is a Y-shaped larger molecule protein 160 kDa bearing two combining side paratopes that bind to a limited surface of an antigen. Epitopes are mostly located on the hydrophilic part of the antigen, and their size is 10 to 15 amino acid residue that can be contiguous on the protein chain, which is linear. The antibody-antigen reaction is a typical reversible bimolecular reaction having rate constant for the forward and backward reaction that are dependent on the concentration of antigen and antibody affinity for the antigen as defined by association constant of antibody for its antigen, temperature, pH and other environmental conditions. Immunoassays are based on the binding and complex of an antigen to an antibody, and people use some physical or chemical means to measure and quantify the antigen-antibody complex. Radioimmunoassay. Radioimmunoassay is defined as it is an immunoassay that uses radio-labeled molecule in a stepwise formation of immune complexes. A radioimmunoassay is a very sensitive in vitro assay technique used to measure the concentration of substance, usually measuring antigen concentration, for example, hormone level in the blood by use of antibodies. Interaction of antigen with antibody are detected by technique called radioimmunoassay. हम इस तरह कहते हैं कि जब एंटीजन हमारे पास है, बाहर से हम एंटीबॉडीज़ ऐड करते हैं तो एंटीबॉडी एंटीजन के साथ कम्प्लेक्स बना लेता है. Radioimmunoassay is used to detect small concentration of antibodies and antigen. एटम के न्यूक्लियस में प्रोटॉन और न्यूट्रॉन पाए जाते हैं. एक एटम है जैसा कि हाइड्रोजन के 3 आइसोटोप्स होते हैं, प्रोट्रियम, ड्यूट्रियम, ट्रिटियम. कोई भी एलिमेंट के आइसोटोप है उनके एटॉमिक नंबर एक जैसे होते हैं, लेकिन मास नंबर क्या होते हैं, एटॉमिक मास मुख्तलिफ होते हैं. प्रोटियम और जो ड्यूट्रियम है ये तो हाइड्रोजन के आइसोटोप्स स्टेबल हैं, लेकिन जो ट्रिटियम है ये जो हाइड्रोजन का आइसोटोप है ये अनस्टेबल है. दीज अनस्टेबल आइसोटोप्स emit particle or energy to reach a stable form. They do so by emitting radiation such as alpha, beta, gamma radiation. Therefore, such isotopes are known as radioisotopes. These radioisotopes can be covalently attached to protein. The radiation produced or emitted by radioisotopes which is attached with antibodies emitted can be detected by the instrument such as gamma counter. Mechanism of radio immunoassay. Step 1. There are many steps explained as step 1 is add the buffer to tube. Step 2. Add known amount of unlabeled antigen to the mixture. These compete for the binding site of antibodies. Step 3. Add the radioactive antigen to the mixture. Now here we use the iodine 125, which is labeled antigen, which is radio isotope. Its half-life is 60 days, which is very long half-life, and it is used as a radio-labeled isotope for detection of antibody-antigen interaction. Step 4. Add the fixed amount of antibody to the tube. In step 5, radioactive antigen is displayed from the antibody molecule by unlabeled antigen. जैसे कि मैंने पहले आपको बताया कि जब एंटीबॉडी एंटीजन के साथ इंटरेक्शन होता है तो हम उसको नेक्ड आई से भी चेक कर सकते हैं. अगर हम चेक नहीं कर सकते उसके साथ हम फिर एंटीबॉडी के साथ हम क्या करते हैं, कोई रेडियो आइसोटोप्स एलिमेंट अटैच कर देते हैं. रेडियो आइसोटोप जो होता है ये अनस्टेबल होता है और रेडिएशन खारिज करता है. ये रेडिएशन मुसलसल जब खारिज होती हैं तो ये जो एंटीबॉडीज़ जो है जिसके साथ रेडियो आइसोटोप अटैच हो चुका है, जब ये एंटीजन के साथ बॉन्ड बनाएगा तो क्योंकि रेडिएशन खारिज हो रही है हम आगे डिटेक्टर लगा देंगे, हमें पता चल जाएगा कि इतना अमाउंट में ये एंटीबॉडी एंटीजन के साथ इन्होंने रिएक्शन किया हुआ या प्रोडक्ट बन चुका है. इन द लास्ट स्टेप द एंटीबॉडी बाउंड विद एंटीजन इज सेपरेटेड फ्रॉम द फ्री एंटीजन इन अ सुप्रानेंट fluid and the each radioactivity of measured compound is determined. लास्ट स्टेप में क्या होता है, जैसे कि मैंने बताया है कि एंटीबॉडी जिसके साथ रेडियो आइसोटोप्स अटैच्ड है वो जब एंटीजन के साथ रिएक्शन करेगी, बाइंडिंग साइट पर अटैक होगी, रिएक्शन होगा, कम्प्लेक्स बनाएगा, इजीली वो डिटेक्टेबल होंगे और लेबल्ड अब ये बन चुका है प्रोडक्ट. हम लेबल प्रोडक्ट को अनलेबल अनटैग प्रोडक्ट से इजीली सेपरेट कर सकते हैं. प्रिंसिपल ऑफ इम्यूनोएसे. Immunology is the study of antigens and their reaction with the antibodies. That is, organism defends the mechanism to foreign bodies by the antibodies. In some cases, an immunoassay may use an antigen to detect for the presence of antibodies which recognize the antigen in a solution. In other words, in some immunoassay, analyte may be an antibody rather than an antigen. Immunoassay analyzer. An immunoassay analyzer is used in hospital and clinical laboratories to run automated biochemical tests to detect the presence and the concentration of substances in the sample. Qualitative immunoassays are often used to detect antigen on infectious agent and antibodies that the body produces to fight them. For example, immunoassays are used to detect antigens on Haemophilus, Cryptococcus and Streptococcus organisms in the cerebrospinal fluid of the patient. Immunoprecipitation is a technique used to enrich or purify a specific protein from a complex mixture using an antibody. Also provides sensitive assay for the presence of a particular antigen in a cell or tissue. It can be used for a wide variety of applications such as protein function and studying. Now we discuss the immunochemical method. It is a process for utilizing the highly specific affinity of an antibody for its antigen to detect the distribution of a given protein or antigen in a tissue or the cell. The first check-home drug test uses lateral flow of chromatographic immunoassay technology, a process that takes place on each of test strips on the test lid. First, the urine sample encounters the drug-specific antibodies that only react to the specific drug or drug metabolites being tested for. Fluorescence immunoassay. It can be defined as immunoassay is a sensitive technique that can be used in the measurement of many compounds such as hormones, drugs, protein, and in the identification of antibodies and in the quantification of antigen such as viral particle and potentially bacteria. In 1944, it was demonstrated antibodies could be labeled with molecules that fluorescent. These fluorescent compounds are called fluorochrome. They have the ability to absorb the energy from an incident light and convert this energy light of longer wavelength and lower energy as excited electron return to ground state. इसमें हम ये देखते हैं, अगर हमने किसी कंपाउंड का स्पेक्ट्रा लिया है, उसकी पीक हमें स्मॉल रेंज में मिली है, सच एज इनिशियल स्टेज में, जैसे कोविड-19 आजकल वायरस है, अगर बहुत थोड़ी मिकदार में स्मॉल कंसंट्रेशन में ये पाया जाता है किसी पेशेंट में, उसका ब्लड हम लेते हैं, अगर उसमें डिटेक्शन नहीं होती लेकिन उसके अंदर वायरस तो है, वो डिटेक्ट नहीं हो रहा, फिर हम क्या करते हैं, एक सबस्ट्रेट यूज़ करते हैं. सबस्ट्रेट वो हो सकता है प्लास्टिक का या ग्लास का, लेकिन अगर हम सिल्वर के नैनो पार्टिकल एज अ सबस्ट्रेट यूज़ करें, उन्हें एक लेयर बना लें. जैसे हम पत्री लेते हैं, पत्री के ऊपर एक छोटा सा सैंपल ड्रॉप रख देते हैं, फिर उसके ऊपर रेडिएशन कंपाउंड माइक्रोस्कोप में रखते हैं, स्लाइड हम कह सकते हैं. तो अगर हम उस स्लाइड के ऊपर थोड़े से नैनो पार्टिकल रख दें, सिल्वर नैनो पार्टिकल या गोल्ड नैनो पार्टिकल, उन्हीं नैनो पार्टिकल के ऊपर फिर हम 1 to 2 ड्रॉप्स या 3 ड्रॉप्स हम ब्लड के सैंपल रख दें, तब क्या होगा, तब हमें फिर हम उसकी क्या करेंगे, एनालिसिस करेंगे, उसका स्पेक्ट्रा लेंगे. वही सैंपल है, जब पहले हमने सैंपल का स्पेक्ट्रा लिया, हमें कोई पीक नहीं मिली, लेकिन जब हमने सबस्ट्रेट ऑफ सिल्वर नैनो पार्टिकल या गोल्ड नैनो पार्टिकल के यूज़ किए, तो वो जो पीक थी, जो बिल्कुल स्मॉल रेंज में थी, आई मीन जहर भी नहीं, शो भी नहीं थी पीक स्पेक्ट्रा के अंदर, तो उसकी एनहांसमेंट बढ़ जाएंगी और इजीली उसकी डिटेक्शन मिल जाएगी. तो हम कह सकते हैं कि पहले हमने जो स्पेक्ट्रा लिया था, ओनली स्लाइड यूज़ की थी एज अ सबस्ट्रेट, तो हमें जो पीक थी वो नहीं नज़र आ रही थी, लेकिन जब हमने सबस्ट्रेट यूज़ किया, सिल्वर नैनो पार्टिकल वो रखे, उसके ऊपर सैंपल रखा, जब हमने स्पेक्ट्रा लिया, फिर उसकी जो पीक्स हैं वो बढ़ जाती हैं और क्लियर हमें नज़र आना शुरू हो जाती हैं. हम इसको कहते हैं सरफेस इनहांस रामन स्पेक्ट्रोस्कोपी. दिस इज़ कॉल्ड फ्लोरेसेंस इम्यूनोएसे. हम कह सकते हैं कि यूज़ ऑफ सबस्ट्रेट के इस्तेमाल करने से पीक्स इन्हांसमेंट हो जाती है और इजीली हम आइडेंटिफिकेशन कर सकते हैं वायरस की, बैक्टीरिया की, जर्म्स की, हार्मोन्स की. दिस इज़ कॉल्ड surface enhanced Raman spectroscopy. Application of immunoassay. Now we discuss the application of immunoassay in food analysis. Immunoassay are biochemical tests that use the binding of antibodies to antigen to detect and measure the concentration of specific molecule. They are relatively cheap, provide fast result, and highly specific due to facts the tests are based on immune responses. Food analysis looks at the chemical, microbiological, physical, and sensory properties of food and drinks. Testing on food need to be done for safety and regulatory purpose, as well as to test for allergic reaction. Traditionally used for clinical study, the use of immunoassay in food industry has grown in popularity over recent years with the rise of fraudulent cases and food contamination. Immunoassays are used in food industry to test the raw materials as well as the final composition of food. They are also used to test for any contamination that may have occurred as well as to test for allergens. Potential contamination can occur during production and transportation. Contaminants tested for include toxin, microorganism, and pesticides. Allergens are tested for both known and unknown allergens. Known allergens are recorded on food label, but sometimes food can be recalled due to accidental contamination with unexpected allergens. Foodborne pathogen testing. Sandwich assays are commonly used for foodborne pathogen testing of E. coli antibodies. A major problem with analyzing foodborne pathogens using polyclonal antibodies in that they have multiple epitopes. Due to this, the specificity of sensitivity can be low and cause false positive results. Monoclonal antibodies are preferred and can be used to test for pathogens such as L. monocytogenes. Bispecific antibodies that recognize red blood cells and L. monocytogenes have been specifically engineered. The bispecific antibodies caused agglutination of red blood cells only in the presence of L. monocytogenes, and agglutination show the red color clump that are visible just by looking with the human eye. Student assignment. Any student have the idea about the advantage of COVID-19 for a biosphere? Discuss the facilities in Pakistan and other countries for treatment of COVID-19 patient. What is advantage of radioimmunoassay? Drawback of radioimmunoassay, and last one is the can we differentiate between COVID-19 MERS and SARS from animal or humans? References. There are many books such as G.D. Christian Analytical Chemistry and Sakoj Fundamental of Analytical Chemistry 2013 9th edition.